Have you ever thought about a scientific experiment as a skyscraper? It might sound strange, but stick with me. If you get the blueprint wrong, the whole thing can just come crashing down. So today, we're going to be the architects. We're going to learn how to draft the blueprint for bulletproof science, the real art of experimental design.
So, let me ask you this. What if a life-saving discovery was built on a lie? Now, when I say lie, I'm not talking about some big conspiracy. In science, a lie is usually just a conclusion built on a flawed foundation. It's an experiment that was, well, unintentionally designed to fail from the start. Let's dig into how that happens, and more importantly, how we can stop it. So, what's the big deal if the design is bad? Well, you get what I call an experimental collapse. And it's so much more than just getting a wrong answer. It's wasted time, it's squandered funding, and it creates these misleading results that can literally send an entire field of research down the completely wrong path for years.
Okay, so here are the usual suspects, the common cracks in an experiment's foundation. Things like not having enough samples, having a biased processing order, or just using poor quality materials to begin with. Any single one of these can be enough to completely invalidate an entire study. So let's figure out how to fix them.
Right, so just like any good building, every solid experiment has to start with a plan. And for us, that plan is the research question. You can't just start building until you know exactly what you're trying to achieve. A fuzzy goal, that just leads to a wobbly structure.
And that question has to be laser-focused. Crystal clear. You can't just ask, what's different in cancer? That's way too vague. A strong question is sharp. It's something like, which specific proteins are more abundant in this type of cancer tissue compared to healthy tissue? See, that level of precision is what guides every single decision you make from here on out.
All right, the blueprint's drafted. Now you need to pick your construction materials. In an experiment, our materials are variables, and understanding them is absolutely critical if you want to build something that's actually going to stand up to scrutiny. First up, we have the independent variable. This is the one thing you are changing on purpose. It's the new drug you're testing, or the different temperature you're exposing cells to, or a genetic modification. Basically, it's the cause. Next, you've got the dependent variable. This is what you measure to see the outcome. So if the independent variable is the cause, this is the effect. We're talking about things like changing protein levels or seeing shifts in cell behavior.
And then there's the one you have to watch out for, the dangerous one. This is the confounding variable. It's that hidden flaw, some outside factor you didn't even think to account for, like a machine's performance slowly changing mid-experiment. This is the kind of thing that can fool you into thinking your drug worked when really it did nothing. Great design is all about neutralizing these hidden threats before they can do any damage.
Okay, so how do we actually lay that strong foundation? Well, there are a couple of non-negotiable principles that prevent those pesky confounding variables from wrecking your whole project, and they are replication and randomization.
This is so important to get right. Let's break this down. Technical replicates, where you're just testing the same sample over and over, all that really tells you is if your machine is working consistently. But biological replicates, where you test different patients or totally separate cell cultures, that's what captures real-world variability. This is absolutely essential. It's how you know if your result is a true biological effect or just some random fluke.
Which brings us to a golden rule. You ready for it? 3. You need at least 3 biological replicates for each condition you're testing. Any less than that, and you just don't have the statistical power to make a reliable conclusion. Seriously, it's the absolute bare minimum for a sturdy foundation.
And check this out. This slide perfectly shows why our second principle, randomization, is a total game changer. Think about it. If you process all your control samples on Monday and all your treated samples on Tuesday, how do you know the difference you're seeing is from the treatment and not just because the lab was, warmer on Tuesday? The truth is, you don't. But by randomizing the order, you make sure that any of those hidden Tuesday effects are spread evenly across all your groups, which completely neutralizes their power to fool you.
All right, foundation's in. Solid. Now it's time for the safety and quality inspections. In an experiment, this means using controls to double-check your work and managing something called batch effects to ensure everything is consistent throughout the entire build.
These are basically your safety nets, your fail-safes. Your negative control is your baseline. It's what happens when you do nothing at all. The positive control is your system check. Does the experiment even work when it's supposed to work?
And then internal standards are like these tiny little monitors you put in every single sample to make sure the machinery is running consistently from start to finish.
Now this right here is one of the trickiest, sneakiest problems in all of science, the batch effect. Let's say you have so many samples you have to process them on different days. Well, you've just created batches. A bad design makes the day itself a confounding variable. But a clever design uses a technique called blocking. You just split your control and treatment groups evenly across both batches. That simple move allows you to mathematically cancel out the whole day 1 versus day 2 effect. It's brilliant.
So you've done all the hard work, the meticulous planning, the strong blueprint, the right materials, the solid foundation, and all the rigorous inspections. What do you get at the end? You finally get to see the finished structure. You get to the discovery.
And this, right here, is the ultimate payoff. All that careful, meticulous work isn't just about avoiding mistakes. It's about building something that stands the test of time. A well-designed experiment gives you a reliable, reproducible result, a genuine insight into how the world actually works. It's how you turn a huge mess of complex data into clear, beautiful knowledge.
So when you think about it, these principles of good design, clarity, having controls, avoiding bias, they're the bedrock of all reliable science. But it makes you wonder, doesn't it? This framework for rigorous, honest investigation, where else could we apply it? What other complex problems in our world desperately need a better blueprint?